Okay, so today we are going to discuss some cells of the innate immune system, and we would discuss first of all the neutrophils and then the macrophages. Although we should have discussed macrophages first, followed by the neutrophils, because of the events that are happening there when the pathogen enters, your body. The first cells that the pathogens would come in contact with are the macrophages. Okay, and then these are the macrophages which actually are going to inform other cells about this attack that some pathogen has entered your body. Okay, and the first cells recruited by the macrophages to the site of infection are the neutrophils. So we would discuss the neutrophils in the coming lecture, but just to briefly tell you about we would discuss the macrophages in the coming lecture. Okay, but to discuss the neutrophils, that what are these cells and what is the role of these cells in inflammatory situation, because these are the cells that are going to enhance this process of inflammation. Okay, and the process of inflammation is you can say that it's a recovery process which is needed for neutralization of the pathogen. That is the process of the immune system that are required for getting rid of the pathogens. Okay, so the neutrophils has a great role in that process. Um, Neutrophils, as you know, are phagocytic cells. And if you remember the previous lectures, I told you that these are granulocytes. They contain different types of granules that we would be talking about. And we call them polymorphonuclear cells because of their peculiar morphology of the nucleus. Okay, there are several lobes of the nuclei in the case of neutrophils. That's why we call them polymorphonuclear cells. The nucleus is not just a single, has not got only a single morphological shape. Okay, it has several, lobes. These granules, which are inside the neutrophils, are very toxic to bacteria and fungi and cause them to stop proliferating or die in contact. Okay, so the contents inside the granules are toxic to different types of pathogens that have entered the body or that has caused infection. And the neutrophils are most numerous. 50 to 70% of all the white blood cells in our body are the neutrophils, and they are produced in great many number, like 100 billion neutrophils are produced in our body every day. Okay, and 50 billion of this number, they are all the times in the blood circulation, and about 3 billion of this population enters the mouth and throat tissues every day, because the mouth and throat tissues are prone to attacks by different types of pathogens, because you are eating different things every day, and these tissues, your mouth and the throat region, is exposed to different types of pathogens. That's why more neutrophils are just infiltrated into this area. So you should just remember that neutrophils are not normally found in healthy tissues. Okay, but they are found in those tissues which have been attacked by some kind of pathogens, because they do not go somewhere if they are not invited there. Okay, neutrophils need an invitation to enter into particular sites or tissues where the infection is taking place, and usually that invitation is given by the macrophages. Okay, if you look at the types and number of receptors on the cells of the innate immune system, there are more than 100 receptors, different types of receptors on the cells of the innate immune system. Okay, and many of these receptors, they recognize different types of pathogens and they respond to different types of pathogens, So generally, if you just look at the number of these receptors recognizing, the number of pathogens, so the number of receptors is far less as compared to the number of known pathogens on these cells. Okay, so how then these cells are going to recognize the pathogens? They are not going to do very specific recognition. Of course, they would be going for some generalized patterns on the microbes to recognize. So, if you remember about pattern recognition receptors, lots of receptors on these cells are the pattern recognition receptors. Just like the toll-like receptors or nod-like receptors or rig-like receptors for different viruses and bacterial species that are present on cells of the innate immune system. So these receptors are present on the macrophages as well as on the neutrophils. Okay, and the neutrophils can neutralize different types of pathogens. For instance, gram-positive bacteria can be taken up by the neutrophils and they just can chop them into pieces and you can get rid of these gram-positive bacterial infections. Similar is the case with gram-negative bacteria. These cells also have proper weaponry to deal with gram-negative bacteria and to just disintegrate the gram-negative bacteria or eliminate them from the body. Okay, so they can do these different jobs with the help of these receptors that are present there. For opsonization also, the receptors are present on the neutrophils. Do you remember the receptors which are responsible for opsonization? I told you in the previous lecture. What are the names of those receptors which are going to bind to C3b, the complement 3b? You don't remember? Okay, so then I should take a quiz in that lecture, I think. Okay. So these neutrophils are different in some respects from the macrophages. For instance, the macrophages, uh, they are residing there in different tissues, okay, and they stay in different tissues for longer periods of time, okay. And the macrophages also are doing many different jobs like, uh, inviting different immune cells to the site of infections, doing phagocytosis of the pathogens, okay, and playing their role in signaling, creating signals for the adaptive immune responses, okay. There are many things that the macrophages are doing, but the neutrophils, they are doing phagocytosis or killing the microbes, but they are not involved in other jobs that the macrophages are doing. Okay, both of them are phagocytic cells, but these phagocytic cells are not doing the jobs that other phagocytic cells like the macrophages would do. Okay, these are specialized cells just to kill the pathogens or the microbes. Okay, that's why if you look at the number of these cells inside the system, as I told you, they are the most numerous inside our bodies. So if you look at the importance of these cells, that in terms of their function, are neutralizing or killing the microbes and taking these microbes out of the system, these are more important as compared to all other phagocytic cells. because they are most numerous. Many of them are just they are taking out more particulate materials as compared to the macrophages from the system. Okay, that's why they are very important. And I call these neutrophils as suicide bombers. You know, because the neutrophils, they are going to die as a result of this fight. Although the lifespan of a neutrophil is very short. The neutrophil can die, its lifespan is maybe a day or two. But even if, when the neutrophil is going to fight and there is some kind of attack by some pathogen, then the lifespan of the neutrophil is shortened. But as a result of that attack, the neutrophil is eventually going to die. Okay, so normally its lifespan is like this one, and they are the first to arrive at the site of infection, which is the hallmark of acute inflammation, as I told you. Okay, acute inflammation and chronic inflammation, these are two different conditions. Okay, acute inflammation has its own hallmarks, or its own certain molecules and cells that are involved in this kind of inflammation, and you can just differentiate between this. Is for ease, you can just remember it like this, that acute inflammation would be the infection that is recent in nature, and chronic inflammation is the inflammation that has taken some time, maybe more than 6 months or so. So that would be a chronic inflammation. Although inflammation is a separate topic, we would discuss inflammation in detail later. Okay. So these neutrophils also have the capability to work under anaerobic conditions. Many body cells are, they are not designed so, or they have not evolved so, even if they were designed initially, because, if you remember about the evolution of cells, so when there was no oxygen, for instance, on the surface of Earth, then the cells that were existing in those times were not using oxygen. They were using alternate sources to produce energy. And when oxygen was introduced, the cells just changed their strategy to use oxygen because it was more efficient to produce energy using oxidative processes rather than going for other processes which were not very efficient for energy production. Okay, that's why since you can say for billions of years, the eukaryotic cells are using oxygen for production of energy rather than going for other things. But these cells are capable to survive in anaerobic conditions as well, because in damaged tissues where there are some problems, the fight is going on, in those tissues the oxygen supply is usually interrupted and not much oxygen is available. So these cells can survive for some time in those tissues, and they have this capability to shift this, their survival strategy for a while, and keep fighting the pathogens for some times and to clear the pathogens. But even if, as I told you, in a normal situation, they still die within hours after entry. If there is no suffocation, for instance, there is availability of oxygen in the infected tissue, even then they would not survive more than a couple of hours. Okay, and eventually when they die, the neutrophils, they would turn into pus, this creamy substance that you see on the when you see some abscesses on the surface of skin. These are staphylococcal or streptococcal infections that cause these different types of skin conditions. Okay, so that creamy pus that you see is mostly the neutrophils there in that pus. Okay, so Where are they coming from? Where are these neutrophils synthesized? They are synthesized in the bone marrow, and they are arising out of the hematopoietic stem cells. And this is the process giving rise to different types of cells. For instance, different types of progenitor cells that are turning into other, if you look at the morphology of the nucleus inside the neutrophil, it is changing with advancing stage of the neutrophil as the neutrophil is evolving here. Okay, so finally, when you have a mature neutrophils with granules formed there, these are filled with different substances that you would see in the next slide. These neutrophils have to stay here for some time, okay? So inside the bone marrow, the neutrophils would stay for 5 days, and then these neutrophils are just, transported into the circulation, the blood circulation. When they come out into the open or into the blood circulation, then the lifespan of the neutrophil is defined that they are not going to live for longer periods of time. But here inside the bone marrow, they would stay here for 5 days. And these batches of neutrophils are then released into the blood circulation, and they then are going to be invited by the macrophages to the site of infections with the help of different chemokine or chemokine factors that are released by those cells. Okay, so normally they are not present in other tissues, but in abnormal situations they would just migrate to the site of infection. Then if you look at the granules, there are different types of granules. There are primary eosinophilic granules, there are secondary granules, and there also are tertiary granules inside these neutrophils, and there are very few mitochondria in the neutrophils because the role of these mitochondria inside the neutrophils, it also is not very well defined. And what are the mitochondria doing in other cells? Do you remember? Hmm? Powerhouse of the cell. So power will be generated in these powerhouses. Okay, but there are few mitochondria, so it means that the power generated by these mitochondria inside the neutrophils, maybe the neutrophils do not require, evolutionarily they do not require a number, the number of mitochondria which are required in case of other cells. They don't require that much power needed as compared to other cells. And the granules that they contain, they have to use up all the granules that they have. Okay, if their lifespan is very short, so they do not have to refill the granules that they also have already have emptied in a situation of infection, or if they have killed some pathogens using these granules, they have secreted the granules and have killed the pathogens, they do not require to make another stock of the granules to refill. Okay, so there is no refilling in the case of neutrophils. But anyway, the granules contain different types of enzymes here. For instance, if you talk about the primary eosinophilic granules, the primary granules include these different enzymes, which include myeloperoxidase, lysozymes, defensins, some neutral proteases like cathepsins, elastases, etc. And inside these primary granules also is a negatively charged matrix. Okay, so on one hand there is a matrix inside this primary granule which has a negative charge, and on the other hand the environment inside this granule is very acidic, it is highly acidic. Okay, and these two things, the negatively charged matrix in the granules and acidity inside the granule, the primary granule that I am talking about, are needed for inactivity of the enzymes. The enzymes that are there inside uh, in this granules should be in inactive form, okay, and they should only be activated when there is some situation, when there is infection, for instance, a pathogen against which they can use this particular enzymes. So this is the strategy of the cell to protect itself using the pH gradient which is there, okay, and the pH would either turn the activity, the enzyme active or inactive, depending upon the situation. Then there are secondary, granules, and these granules contain lactoferrin, for instance. These also contain lysozymes. These also contain NADPH oxidase. Okay, so the lactoferrin, which is there in the secondary granules, it competes the pathogen for metal ions, for instance. Okay, so it can kill the pathogens by this kind of competition. Okay, this is a the pathogens are competing for ions in this situation. The lactoferrin is going to take up some iron which is needed for the pathogen. Okay, so not much of that is available for the pathogen. That's why the pathogen is at a disadvantage in this situation. Okay, and it's going to die. Then there are other enzymes, lysozymes, or NADPH oxidase, which is very important here for the function of the neutrophils. Okay, because this NADPH oxidase is going to produce superoxide radicals, and these superoxide radicals are converted into hydrogen peroxide, and hydrogen peroxide uh, formation takes place with the help of an enzyme which is already there inside the neutrophils, and that is the superoxide dismutase. Okay, what is the job of hydrogen peroxide? Hydrogen peroxide is very toxic. Have you ever seen hydrogen peroxide sold on the market inside the medical store? People are using hydrogen peroxides for different purposes. For instance, if you are just taking care of the wounds, or some people just would mix up hydrogen peroxide with soda to brush their teeth, because this is very potent in killing bacteria, even those bacteria that are hidden in this yellow tissue, yellow thing inside your mouth, that calcium built villas on top of your teeth. It's not allowing any of these different types of chemicals or something that you are using to just get rid of them, because it's not accessible. The bacteria are hidden inside, they are not accessible to the drugs that you are using against them. Okay, but these things actually are helpful. Hydrogen peroxide based solutions, or you can say, you can just mix some hydrogen peroxide with soda, and then you can use that for a couple of days, and that also can help with killing the bacteria that are hidden there. So this is very toxic to the cells, the host cells, as well as cells that are not belonging to the body, that are the pathogens. Okay, the tertiary granules, the third type of granules, they contain gelatinase, and this gelatinase also restricts the growth of bacteria by sequestering iron. Okay, more like the secondary granules. So these are different granules. The first thing that these neutrophils are going to do when they engulf the pathogen is to make use of the primary granules. Okay, they are going to neutralize or disintegrate or kill the microbe by releasing primary granules, where when the pathogen is engulfed, it would be kept in a phagosome, and in that phagosome, these granules would be just, released on the pathogen inside the phagosome. And these granules, the primary granules that I'm talking about, are going to, there are different types of enzymes there that are going to kill that microbe or turn it into, pieces. Okay, and that can happen immediately after ingestion of the pathogen. It would just take a day or two for the neutrophils to just, mount this response and get rid of the pathogens or bacteria, etc., that have entered the body. And after that, if it doesn't work and the pathogen still is persisting there, and the neutrophils are coming and fighting with them, the neutrophils have other options as well. So they are also going to make use of these options in order to use this weaponry to secrete granules into their immediate, premises or environment, the microenvironment. And those granules which are going to, compete, for instance, the ions, the irons, for instance, are different ions, and these granules also contain different types of lysozymes. This contains NADPH oxidase, which is going to give rise to hydrogen peroxide. So the phagosome then is also going to finally be fused with the lysosome, which is another, factory of very dangerous enzymes inside the neutrophil. Okay, in phagolysosome, the phagosome and the lysosome finally, when they fuse with each other, they would have all these reactive oxygen species, the hydrogen peroxide produced by these enzymes, and there are lots of acid hydrolases that are going to finally, chop that pathogen into smaller pieces. Okay, so the pH, when all these actions of the primary granules and the secondary granules are taking place, the pH inside this granule is going to turn into basic. It would rise up to 8, okay, or more than 8. And after this, function or this, killing of the microbe is finished, then the pH is going to drop down. Okay, and it will become neutral, or at pH 7, these enzymes again are not going to work. Okay, what is the fate of the neutrophils finally? The neutrophils, after doing this particular job, is going to die. Okay, that's why I told you earlier that it's a suicide, bomber. It's going to die as a result of this encounter.